Happy Friday, everyone. This is going to be the start of the endocrine section. This is the last section that we're going to have for the exam. We'll get it started today, and then we'll finish it on Monday. So we're going to start today with just understanding what the endocrine system is. And it's an intracellular communication system. It is our ability to send a signal from one cell to another. We talked about gap junctions sending cytoplasmic cells, but this is actually going to leave a cell and go to another cell, or sometimes itself, but from the outside. So we can say that there's 3 different signaling types. If we release hormone from one cell and then it binds back to the same cell, that's an autocrine signal, because auto means self. Paracrine means nearby, so we can release a signal from one cell and it will diffuse locally to its neighbors and bind and cause a response. But then we also have endocrine, which is going to be in the blood. So for anything that's further away than a diffusible distance, you have to put it in the blood and then let the blood take that hormone all the way through the body to whatever system it has to go to. We have receptor characteristics. The receptors are the important part of this story. You can have all the hormone you want in the world. If you don't have a receptor for it to bind to, you will not get a response. So we're going to look at the receptor and how it has the specificity for its hormone. Some have higher specificity than others. So some can actually bind similar hormones. It's going to have an affinity, which we call a stickiness. That's how well it can hold on to that hormone once it's bound it. That didn't seem like correct grammar, but once it has it bound, whether or not it's going to hold it for a long time or not. And then we'll talk about dissociation constant, which is really a quantifiable measure of that affinity. Instead of just saying sticky, we can put a number on it. We can look at what concentration of hormone we need to be able to fill 50% of our receptors. And that leads us to the law of mass action. Don't panic, we're not going to get deep into this. This is chemistry, which is my least favorite subject, so you know we're not doing that. You won't have to even calculate anything here. We just want you to understand this principle. And I just wanted to point out too that anytime you see an ion, an atom, a molecule, and it's surrounded by these square brackets, that means concentration. So we're saying the concentration of the hormone plus the concentration of the receptors. How many receptors we have, how much hormone we have. Add those together, you can get hormone binding in this hormone receptor complex, but it's not going to stay permanently. That hormone will release from the receptor, so you can move this equation back the other way. And law of mass action is just saying that eventually this will reach equilibrium, so that the rate that these hormones bind to the receptor will be equal to the rate that they dissociate from the receptor. Okay. Then we're going to talk about hormone types. Receptors are the important part of the story, and we'll talk about exactly how they're causing these responses, these signals on Monday. But first we need to understand the differences in the types of hormones that we can have, so we can see why we have different receptors. So we can have peptide or protein hormones. Okay, a peptide is a small protein. That's a relative term. That means maybe 100 to 200 amino acids in it versus thousands that you might see in a typical protein. So they're considered to be large. Although proteins and peptides can actually be hydrophilic or hydrophobic, the ones that we use as a hormone are typically hydrophilic. They're typically charged. Then we're going to also see that we can use amino acids, like individual amino acids, not 250 or 1,000, but just one amino acid, and we can modify that into a hormone. So these are much smaller. We could take tryptophan and turn it into serotonin. It's a really important neurotransmitter. Most of you have probably already heard about it. We're not going to talk about it in this class. We don't get into that. So psych classes, neuro classes, you will talk about this a lot. Here, just know that it comes from tryptophan. Catecholamines, which are your dopamine, your epinephrine, your norepinephrine, we're going to talk about these quite a bit. These have a lot of physiological functions throughout the body. These are all called catecholamines, and they're coming from tyrosine. Okay, so you change a tyrosine to a dopamine, then you can change a dopamine into an epinephrine. Actually, you change it into norepinephrine, and norepinephrine into epinephrine. Tyrosine can also make a thyroid hormone, but it's going to have very different characteristics from the catecholamines, so we'll talk about that. And then this is a really important class of hormones here, steroid hormones. They're coming from cholesterol. So you take cholesterol, fat, and you can turn them into all of these different types of hormones. You've got all your sex hormones we'll talk about in repro. You've got cortisol and corticosterone we might talk about a little bit, mostly stress response. Aldosterone is going to come up in kidneys. It's really important for modulation of our kidney function. And then calciferol we don't usually talk about, but I'm going to talk about it in a story today. Calciferol is also called vitamin D. It's really not a proper name for it because it's not a vitamin, but it's what most of us recognize. Vitamin D, or calciferol, is really important in helping to absorb calcium. So we know how important calcium is. We know that if we don't have enough calcium, we'll strip it from our bones. So having vitamin D to be able to absorb that calcium is a really important part of the story. And that's going to lead us into the story I wanted to get to yesterday. I didn't have time. I thought I was going to drop it, but then I decided with recent events that I want to add this in anyway. So I'm going to give you an abridged version. It would have been abridged anyway because it's really Walren's story, so he knows all the details. But this is going to be a shorter version, and this is going to be the story of melanocytes. Okay, so we talked about the epidermis, and we said that there were these minimum of 4 layers. Just going to draw a simple epidermis here. And we'll have our basal lamina in here. So we know now that this is the stratum basale. So first I told you there was keratinocytes, then I added stem cells in, now we're going to add a third cell. So we do this a lot. We give you kind of the simplified version and then just add things in later once you've got the foundation. So here's your melanocytes. Okay. Melanocytes produce melanin. And melanin is a pigment. Just like henna, this pigment can be taken up by keratinocytes. So the melanocytes will release this, and it will get picked up by your keratinocytes. You constantly have to make more because you shed these keratinocytes off. So you've got to get more pigment in them. But the story of the melanocytes is a fascinating look at evolutionary pressures in our history. Because when man first started into Homo sapien from our primate brethren, we were in Central Africa in the rainforests. We had fur on our bodies. We didn't quite walk upright yet. Time went on. We started walking. Eventually, we moved out of the rainforest and onto the plains. Better hunting. But now we've got a ton of fur, and we're running around trying to catch animals. And we're in Central Africa with the sun beating straight down on us because this is at the equator. This is really hot and really bad. So there's a selection pressure now for people with less fur who can stay cooler and not have heat stroke in the middle of their hunt to reproduce more. So eventually over time we end up with no fur at all. We have hair. Some of us are fuzzier than others, but we don't have a layer of fur. That helped keep them cool, but it led to another issue because we know that sun carries ultraviolet light. These photons of ultraviolet light have a lot of energy, especially UVB. So much energy that this can break ring structures. So we know this now. We know that we have to use sunscreen to protect ourselves from this UV because this is going to damage DNA. We have a ton of rings in our DNA. But that's also a long-term problem. Cancer is something where you get the damage now, you see it decades from now. Early man didn't survive that long. Cancer wasn't a concern. But that's not the only molecule that they have that has ring structures in it. We have lots of proteins that have rings. So having all of this UV breaking all of those proteins caused a lot of problems. And so there was a selection pressure now to have more shielding from that UV. And we could do that by increasing melanin, because melanin acts like that shield. You can think of it as sunscreen for the cell. Okay, so over time, they built up a sunscreen of about SPF 50. You know, nicely shielded from this UV. But all of that pigment shows through, just like henna would. And you get this darker skin tone in the process. Hundreds of thousands of years go by, and now people are starting to move. They're migrating. Populations start going north and heading either east to Asia or north into Europe, northwest into Europe. And now they're at higher latitudes where the sun does not come straight down, it comes down at angles. So they have less UV exposure here. It's also colder, which is kind of a problem because they got rid of their fur, but hey, they can just go steal fur from other animals and wear it. And there's an advantage here because now if they get hot, they just take it back off again. But while they are wearing this fur, they get even less sunlight. So you think, well, that's good because UV bad. Except that we do sometimes need this UV. We said we had these steroid hormones that are based on cholesterol. We have to make changes to these cholesterols to get them into these other forms. And so when we're making calciferol or vitamin D, the way we do it is to break a ring in cholesterol. UV is great at breaking rings. So UV is a great way for us to make cholesterol into vitamin D or calciferol. We can also eat vitamin D. I mean, we sure do now. If we're eating any processed foods, they're probably adding tons of vitamin D in there. Early man had vitamin D because they ate animals. Other animals do the same process. They make vitamin D, we eat them, we get it. But now we're not making as much in these northern populations because they're not getting as much UV exposure. So there becomes a selection pressure to move the other way. So now all of a sudden, these populations have to decrease that melanin to allow more of that UV light through so they could still make vitamin D. This continues on for thousands of more years and everything's going fine until society has another major shift. They start moving from hunting to agriculture. They start planting a bunch of grains, and now they're eating all these grains, and the grains don't have vitamin D. So they're getting UV from, or cholesterol to calciferol from the UV, but they're losing a lot of their source of dietary, so they need to make more. This mainly happened in Northern European groups. The East Asians still had enough dietary vitamin D that they didn't have the same pressure. But now you've got another round of mutations. Okay, so you've gone from SPF 50 to like 30 in the initial round to Northern Europeans are now down to like SPF 10. Okay, if you're like me, I'm a mutt, but I'm mostly a Northern European mutt. We're pasty white. We don't even have any melanin left at all. If I go out in the sun, I don't tan, I turn to a tomato. Eventually I get freckles, but that's about it. Okay, so this is an awesome look at how evolutionary pressures can change something that we can see. We know that we've had these pressures before, but this is something you can like just look and see what happened. It's also really neat because you can see how it pushed in one direction and then changes in location and diets pushed it again in another direction. So now we've got these populations that have spread out all over the globe, and we've got this wonderful diversity of amounts of pigment, which leads to this beautiful array of skin tones. And I wish I could say that as much as I love this story, that it was all rainbows and puppy dog tails. We know it's not, because somewhere along the line, people decided that these levels of pigment could actually be used to measure or judge people's intelligence, their character, or their worth as humans. And it's just ridiculous because it's just pigment. It is mutations based on location and diets. Having more or less pigment does not make anyone more or less a human being. Okay. Thank you. Sorry, I felt like that was really important right now. But that is going to end our skin story, and we can move to endocrine, unless anyone has any questions. Okay. So endocrine systems. Intercellular communication. When you have an endocrine system, you have to have one cell that sends a signal. This sending cell is going to release hormone. Again, this is going to be something that we actually have to exocytose from the cell. We have to put it out into the outside world and send it elsewhere. So this is going to be exocytosis of your hormone. In fact, instead of secrete or release, I should say secrete. Because of this, because we always have to be able to secrete this hormone, that tells you that anytime we're talking about a hormone, this is going to be made on a membrane-bound ribosome. Okay. Now you've got to have something to receive the signal. So you have your receiving cell. This is going to bind to that hormone. And cause a response. Again, we'll talk about the array of responses that we can have to this signal on Monday. But for now, just understand that we have to have a receptor. We can have all the hormone in the world. If there's no receptor to respond to it, it doesn't do anything. So this receptor, we say this again and again, it's always about the receptor. Now we can have varying degrees of migration of these hormones. We can talk about a cell that's using autocrine signaling. So again, autocrine is self. In this case, this cell could release this hormone. And the receptor for that hormone will be on the same cell. So this hormone could diffuse over and bind. We would say that this cell is both the sending and receiving. On the other hand, we could have a neighborhood of cells. So we've got this nice epithelial layer here. This is going to be our sending. So it will secrete its hormone. And cells nearby may have a receptor that can bind to it. So as it diffuses over here, so these are receiving. Now, just like with oxygen, all molecules really have a limit to the ability to diffuse. So when we're trying to diffuse hormones, it can only get so far before it gets degraded, before it gets reabsorbed, or it's just too diluted to actually function anymore. So if you need to send it further than just the neighborhood cells, you need to use an endocrine system. Oops, I forgot to write, this was paracrine. So in an endocrine system, you're going to release hormone, and a nearby blood vessel can pick that up. Now this blood vessel is going to go all throughout the body. We have a closed system. We've got blood everywhere. It's going to flow through the body. It'll diffuse out into spaces. Most places it's probably not going to be able to bind. It's not going to have receptors. It has to find the portion of the body that actually has the receptors it's looking for. But when it gets there, when it finds its target cell, this target cell is going to have a receptor on it. And so you'll have this other blood vessel that's close to this cell. And you'll have some hormone diffuses out and binds. I'm trying to make a little point here. And that's that when you put these hormones out and they're in a small area, they seem like a high concentration. But once you put them into the blood and they diffuse throughout the body, they become very diluted. You can picture this as using a really big aquarium, and you take a bottle of like food coloring and you pour it into one corner. So you could see in that one corner that you've got plenty of molecules. But when you come back an hour later, you may not even be able to see that dye anymore because it dilutes out through the entire aquarium. So we would either have to put out a crap ton of this hormone all the time in order to get high concentrations to all of our receptors, or we have to make our receptors so sensitive that they can deal with these lower concentrations. We took the latter route. So a normal hormone concentration is typically going to be in the range of 10 to the negative 9 to 10 to the negative 12 molar. Very small concentrations. Okay. All right. So let's look at the receptors themselves. We have receptor characteristics. And the first one we want to talk about is the specificity. Specificity. Yeah. This is the idea that a hormone receptor will be very specific for a particular hormone, but you can have some variation. So if you had a receptor it will not look like this, of course. It's 3D, it's much more complex, so its structure is going to be a lot more specific. But this is a good example for saying if we had a hormone, let me label this as a receptor. Okay, so we have hormone 1. This is the hormone that's meant to fit. We can kind of look at it and see that that's going to just slide in there real nice. Okay, it'll bind, it'll stay. What if we had another hormone that looked like that? Versus a hormone that looks like this. You wouldn't expect hormone 3 to bind at all. It just doesn't even have a similar shape. There's no way it's getting in there. But hormone 1 is definitely going to bind, so this one's A-okay. We'll say no on that one. If hormone 2 lined itself up in just the right way, and that looked like a parallelogram. That was meant to be a square. Okay, I'm just making a bigger mess. I'm gonna say theirs. So just turn the square into a triangle, and maybe you can shove it in there. If this receptor is highly specific, we're gonna use orange here, then this would still not fit. How do I want to do that? If it's highly specific. If this receptor has a slightly lower specificity, then that one might actually work. You'll typically see this with your hormones that are similar in where they're coming from. Okay, so we know that we can make these catecholamines, we can make dopamine into norepinephrine into epinephrine. They're typically only maybe a group apart. You take a methyl off, you add a hydroxylon. They might be similar enough in shape that they can bind a not quite so specific receptor. Okay. I like to think of that video where they had the kids' toy with like the cutout of the square and the triangle and the circle and the moon. And he picks up all the pieces and he puts the square in the square, and then he picks up a star and he puts it in the square, and he picks up everything and he puts it in the square, and about had an aneurysm. That is low specificity. If that had actually worked right, it would have been high specificity. Now, even if you can get this hormone to bind, if it is low enough specificity that it will pick up a slightly different hormone, it's still not going to bind as well. It's not going to stick quite as well. This 3D binding site has an exact match for its hormone. It will have good affinity for its hormone. It will have low affinity for another hormone that's similar but not quite the same. So when we talk about affinity, we say that stickiness. But you can also think of it as how well it can hold the hormone. And we can measure this. We can measure this as a dissociation constant, or KD. So the definition of KD is the concentration of hormone necessary to bind half of the receptors. So let's draw this so we can actually visualize this. If we had a group of cells and they all had our particular receptor on it, and of course you probably have hundreds, thousands, hundreds of thousands of receptors for any given hormone, I'm going to draw 4. Worse, I'm going to use numbers that are nice and round. Okay, so, oops. No wonder I couldn't get it to fit. I can't draw. Oh, there we go. We know that hormone concentrations are typically super, super low. But I deal with really small numbers about as well as I deal with really big numbers. That's too many zeros for my brain. So I'm just going to give you nice even numbers and recognize that we would not actually ever have hormone concentrations that look like this. But if we take these receptors and we do a test with hormone 1, and we see how much concentration of this hormone it takes to bind 50% of these receptors on average, keeping in mind that they will bind, they will release, they can rebind again, new ones can come in, this is constantly changing. But on average, if 50% of these receptors are bound at a certain concentration, here we'll say that's at a hormone concentration of 1 molar. In other words, if you put 1 mole of this hormone in, you can bind 50% of the total receptors that can bind it. If you then added more, 1.5 moles, you'd increase the percentage of receptors that you could bind. You decrease this concentration, you wouldn't be binding quite as many receptors. Okay. This is your KD. 50% concentration, that tells you that your KD is 1 molar. If we ran this experiment again, same receptors, but now we looked at hormone 2. We're going to say that hormone 2 can actually bind. This receptor wasn't terribly specific, but it's not going to bind as well. We know that. So now to get 50% of these receptors bound, the concentration of this hormone, think it's going to have to be higher or lower? Higher, perfect. It doesn't bind as well. It lets it go all the time. So at any given moment, you're going to have fewer of these hormones bound to the receptors. We could say, just making this up, that this is going to be 2 molar. So again, this means you've got a KD of 2 mole. This KD is bigger than this KD. Okay, so this is like a high KD. This is a low KD. Of course, if these were actual numbers for your KD, they would both be excruciatingly high. But for our example, this is low KD, that's high. And we know, just from thinking about it, that these hold well, so they have more stickiness, that's a higher affinity. And that because this is not as sticky, it doesn't hold as well, this is a lower affinity. Hopefully you recognize that relationship. If not, I'll spell it out. High affinity is measured as a low KD. Okay, I told my last section, I'll tell you. I would really love for you guys to understand the why of this, but if you walked out of here and just memorizing this, I might cry, but you will do just fine on any questions about this on the exam. That's your takeaway. So any questions on receptor characteristics? Yes?
Can you give an example of like why or when this would happen?
What would happen?
Like a different hormone binding to the site.
Okay, so when would we have something different that binds to the site? We're going to talk a lot about epinephrine and norepinephrine, and we will have specific receptors. So if we said that this receptor was an epinephrine receptor, this would be epinephrine. I'm just going to write epi for epinephrine. Okay, this has no problem. It has a low KD, high affinity, because that's the hormone that's meant for it. We can also have norepinephrine, norepi, that is very, very similar And it typically has a lot of the same functions. We use them for a lot of the same things. So some of our epinephrine receptors might be very specific and they will only bind epinephrine. Some of them might be slightly less specific and they can actually bind to norepinephrine. It's going to have a lower affinity, which means you have to have a higher concentration of norepinephrine. Essentially what you're saying here is that we have a normal concentration that can activate this, and we can sometimes use another molecule to do it, but it has to be in much higher concentrations than normal. You can imagine that that might be an emergency situation, okay, because that might be the only time that you would have that high of a concentration of that other molecule. Okay, so maybe there's a problem and you're not producing enough epinephrine, but if you could produce enough norepinephrine, you can still get whatever that signal was done. Do you have more questions? Yeah.
I do. So for paracrine, does it have to go outside into the extracellular matrix, or can it go in between cell membrane to cell membrane through like a vesicle or something? So does it have to go like completely outside of the cell?
So if it went out, it can't go out in a vesicle. This is a cell, so a vesicle would actually merge with the membrane and exocytose. You would never have vesicles out here in the extracellular space. I should not have said that. Never say never. There's an exception, but for our purposes now, we do not put vesicles out here. In order to get it to another cell without having those gap junctions to connect them, you have to go outside the cell, just as a...
And then for biocrine, can a cell make a hormone that's completely sitting inside the cell membrane because it's sending a signal to itself? Or does it have to leave and then go to a receptor that is on the outside of the cell?
Okay, so does autocrine always have to go out, or since it's going to the cell, can it just stay inside and signal? The way that these work, and I'm specifically drawing transmembrane proteins here, transmembrane receptors that are going to bind hydrophilic hormones. In this case, it has to go out and signal because it's this particular transmembrane protein that's going to cause a series of events. You have to trigger it to get that to happen. If we're talking about hydrophobic, which actually bind to cytoplasmic receptors, I'm not sure. I actually never thought of it before. We usually use the transmembrane hydrophilic story as example, so I'm not sure about that one.
Okay.
Are stickiness and affinity like the same thing?
Stickiness and affinity are the same thing. Stickiness is how you can describe affinity.
Oh, okay, thank you.
Yes.
So if receptor or the hormone 1 and hormone 2 are binding to the same receptor, is there going to be any difference in like what happens after they bind?
If you have epinephrine and norepinephrine binding to this receptor, does the fact that you're using two different hormones have any effect whatsoever on the response that's going to happen? No. It is always about the receptor. These receptors, we'll see, there's only a couple of different categories. While the receptor shape itself might be specific for a particular hormone, every receptor within that category will have the same response. Okay. Yes?
So for the concentration of like hormones in just all of them across the board, is it like specifically endocrine that has the 10 to the negative 9 and negative 12 molar, or is it just all of them?
Yeah, I got asked this last section too. So we talk about how we release it, and we release it in high quantities, but then it just diffuses. I would assume that when we're talking about these concentrations that it's after the full diffusion, okay, because that's our actual concentration at binding. I would also assume that if you're just diffusing and it's not going to lose as much concentration, that you would have released less to start with to have that same low concentration at binding. Anyone else? Yeah.
For high affinity, it means low dissociation constant, but does high affinity mean high specificity?
Does affinity and specificity relate to one another? Probably yes. If you have a specificity for a particular hormone, if you have the perfect 3D pocket shape for it, I would expect that it's probably going to stick a little longer than average. But it's not a perfect correlation. Even if it fits really nicely, it may not be strong bonds. It just may not have a lot of bonds between it to hold it. So generally speaking, yes, they're correlated, but not perfectly. Okay. Let's talk about hormones. So we're not going to go into details of these hormones and look at the structures of all of them. We're just going to talk in general terms about what kind of hormones they are, whether or not they're hydrophilic or hydrophobic, and therefore whether they're going to bind to transmembrane or cytoplasmic receptors. So we could talk about our hormone. We can talk about what it's made from, whether it is hydrophobic or hydrophilic, and then as a result, whether it's going to bind to a transmembrane receptor like we've been drawing, or a cytoplasmic. So we said our first one was going to be these proteins and peptides. And we said these are large, charged, and hydrophilic because they're going to have at least 100 and possibly thousands of amino acids in this. But we're only going to use ones that actually have this charge. And so therefore hydrophilic. This is coming from amino acids. And so hopefully you recognize that the hydrophilic has to be transmembrane, that hydrophilic cannot move through the plasma membrane, that it can't get through those fatty acid tails, and so that it's going to have to actually use those transmembrane proteins. So anytime we talk about hydrophilic, we're going to be talking about it binding to a transmembrane protein. And then we said we had serotonin. There's no rhyme or reason to these colors. I'm just trying to differentiate them, so don't worry about finding the right color. Serotonin came from tryptophan. This is just one particular amino acid. Tryptophan is hydrophilic. It's polar. It has a ring structure. When you turn it into serotonin, it stays hydrophilic, which means that's transmembrane. You can also make these catecholamines. Those are your dopamines, epinephrines, and norepinephrines. Those all came from tyrosine. This is also a polar amino acid, has a ring structure. It will stay hydrophilic after its conversions. So it's transmembrane. But then we get to thyroid hormone. Thyroid hormone was also made from tyrosine. But the process of making it into thyroid turns this hydrophilic molecule into a hydrophobic molecule. Because it's hydrophobic, it doesn't want to be out in the extracellular space. It's not going to be binding to this transmembrane protein. It can, however, pass right across that phospholipid bilayer. And so we can bind these to cytoplasmic receptors. Last but certainly not least, this is one we're probably going to talk about a lot. This is going to be the steroid hormones. I won't list them all out here. You should have written them down there. I will point out that when you're looking for what kind of hormone it is, steroid hormones tend to stand out. A lot of them actually have sterone in their name. Okay, I always think of that as meaning steroid hormone, sterone. If they don't, which unfortunately we have a couple that don't, they'll have this all, estradiol, cortisol, cholesterol. All right. We're not just going to dump random hormones on you and expect you to just kind of tease out from the name whether it's hydrophilic or hydrophobic. It's going to be activating a transmembrane or a cytoplasmic receptor. There's a couple of names that you should probably be familiar with because we'll talk about a lot: epinephrine, norepinephrine. Know your sex hormones. I mean, those should just be automatic. Otherwise, we're going to give you some kind of hint. We'll tell you something about it that should be able to make it obvious that it's either hydrophilic or hydrophobic. But we know that since these are coming from cholesterol, and that's a fat, this is going to be hydrophobic. All of your steroid hormones are hydrophobic. Therefore, they're all going to bind to cytoplasmic receptors. Okay. Questions? None. There we go.
Are we supposed to like memorize this table, or help people to understand this for the exam?
I would be familiar with I would actually know the table, okay, because I didn't put all the specific hormones on there. Just know the categories and know which ones are hydrophobic and hydrophilic. So essentially, your proteins and peptides, and then most of your amino acids are going to be hydrophilic. Your exception is going to be your tyrosine to thyroid hormone, and then any of your steroid hormones. All right, I'll see you guys on Monday. Thank you very much. Come on. Estrogen is estradiol. In humans, it's estradiol.